Anvita. First question. Unicellular microscopic organism were Leeuwenhoek. So, Sandana, you have your mic is on. So, unicellular microorganism, Leeuwenhoek, and this is given in the NCERT first chapter, as you can see here. Anton von Leeuwenhoek first saw and described live cell. So, this live cell was unicellular organism.
Good afternoon.
Good afternoon, Chandana. Okay, move to the next question. Chandana, Swan, 1839, proposed that the body of the animal and plant are composed of cell and cell product. His idea is hypothesis. So this topic is given here, cell theory. There were two scientists, 1838 Skeleton, who examined large number of plant and said all plant are made up of different type of cell, and these cell will form tissue. So he gave also idea of the tissue. Same Theodore Swan, 1839. He studied animal cell and reported cell have thin outer layer. Today we call it as plasma membrane. So he introduced the idea of plasma membrane. Swan. He concluded based on his study on the plant tissue that presence of the cell wall is the unique character to the plant. So cell wall idea he also introduced. Cell membrane, cell wall in case of plant. On this basis, Swan proposed the hypothesis. On this basis, Swan proposed the hypothesis that animal and plant body composed of cell and cell product. Done. Third question. Marinder, slime layer or a capsule is a modification of which of the following layer of the cell envelope of the prokaryotic cell? Marinder, am I audible?
Yes, sir.
Glycocalyx. You told कि sir क्या आप explain कर सकते हो तो I'll explain this topic. So when we read about the prokaryotic cell, there are characteristic feature in the prokaryotic cell, and one of the characteristic feature is glycocalyx. Okay, and here in this topic we have this part. Most prokaryotic cell, particularly bacterial cell, have complex cell envelope. Cell envelope have tightly bound three-layer structure, where you have outermost glycocalyx, then cell wall, and after that plasma membrane. Each layer have different function, but they act as a single protective unit because they protect the bacteria ultimately. Bacteria, on the basis of the difference in the cell envelope, like gram positive and gram negative bacteria have different cell envelope, and when we color them, they respond in a different manner. When staining procedure means color them, when we color them, so they respond in a different behavior, and this was explained by the Gram, who said so. The organism that take Gram stain is Gram positive bacteria, and bacteria that do not take Gram stain is Gram negative bacteria. So bacteria are two types based on the cell envelope and staining process. And these are very popular. You will study in MBBS also, like in microbiology, you have a lot of Gram positive and Gram negative bacteria. So glycocalyx have different composition and thickness. So this question can be asked: what is a different version of glycocalyx is present? It cell can vary in the composition as well as thickness in its glycocalyx. If it is loose sheath, slime layer, glycocalyx only. But this glycocalyx can be present in the two form: loose sheath slime, very thick and tough layer capsule. And as name suggests, capsule will protect the uh, bacteria more than the slime layer. Any doubt, Barinder?
No, sir.
Move to the next question. Jordan. Incorrect statement regarding the centriole.
Present in animal cells, true.
Hmm.
Helps in spindle formation also. Present in higher plants, it's false. Because
Hmm.
here the cell plate forms for instead of the furrowing part. That's
Uh,
also another point
so
in
we
terms
should
of
know these
difference.
four things. It is present in the animal cell. It help in the spindle formation. It means made up of the microtubule, and it is present in the higher plant. So if you look at the introduction part given about the centriole, it is the uh, one thing is this: animal cell contain non-membrane bound organelle. Non-membrane bound organelle, which is known as centriole, and centriole help in the cell division. Give me a minute. Yes, guys. So one thing is given here. Second thing is given in the eukaryotic cell. Second thing is given in the eukaryotic cell. Example, this. Animal cell have centriole, which is absent almost in all plant cells. So this was the statement given here. Okay. Third, you should know about the centriole because centrosome we have in our portion. This. This centrosome is an organelle and made up of two cylindrical structure centriole. One centrosome made up of two centrioles. These two centriole are surrounded by amorphous pericentriolar material. So there is material present around the centriole. It will protect it and help in the spread. Both the centriole are perpendicular to each other. Example, one centriole is like this and second centriole is like this. Both are perpendicular to each other. Huh. And in which each, remember, not dosa देखो हाँ इनमें देखो in which you have each have organization of the cartwheel. Each have organization of cartwheel. Individual will have cartwheel structure. How? uh, I will show you cartwheel structure of centriole. Individually it look like cartwheel, one centriole, not both together, and made up of three unit triplet. Okay, most of the things are same as cilia and flagella. So next, they are made up of nine evenly spaced peripheral fibril made up of tubulin protein. So there are peripheral fibril outside five, and how many? Nine, equally spaced. Each fibril is triplet. Adjacent are linked with the linker. Central part of the proximal region to the centriole is made up of protein known as hub. Even in this case, this word proximal region is important. Central part of the proximal of the centriole, just in the front part of the centriole, at the center is known as the hub. and this hub is connected to all fiber with the help of radial spoke. So it's like एक दो 1 2 3 1 2 3 1 2 3 1 2 3 1 2 3 1 2 3 1 2 3 like this there will be 9, and all will be equally spaced. At the center part it will be hub made up of the protein, and the center part will attach to all these with the help of radial spoke, and they are attached to each other with the help of linker. With the help of linker they attach to it. Okay. Same centriole form the basal body. Same centriole form the basal body for the cilia and flagella. It means cilia and flagella arise from the basal body, and basal body arises from the centriole, and spindle fiber give rise to the spindle apparatus during the cell division in the animal cell. So we know in animal cell there is cell division, and the structure responsible for the cell division are the spindle fiber, and these spindle fiber arise from the centriole. That is the significance of centriole. And if you conclude the same thing here in the diagram, it is present in animal cell, correct. It helps in the spindle formation, spindle apparatus, correct. It made up of tubulin protein, microtubule, correct. This is it. Give me a minute. So move to the next question, Tuhina. Correct match. Tuhina.
So
Yes, Jordan.
you want to know whose turn was it? That's why I asked.
No, Tuhina. Is she
So
there?
the correct match will be 2, right?
Correctly match.
Yes, sir.
Is there an option? No. Mitochondria is not protein packaging because mitochondria function is energy.
Yes, sir.
RER not
Energy production.
photosynthesis. Uh, Golgi
And
apparatus is wrong. So
it
SER is
packages. Yes, sir.
detoxification. Okay. So in this you can go through the detoxification, by the way, is given in the summary. See here. Endoplasmic reticulum contains tubules or cisternae, rough and smooth. Endoplasmic reticulum help in the transportation of substance, synthesis of protein, lipoprotein, and glycogen is not given in the NCRT. Normal lipoprotein is not given. Transportation of the substance is not given. There was detoxification I have read in the NCRT. It was that maybe we'll do later. We will write in the SER. Here you can write detoxification. Angur, detoxification in case of smooth endoplasmic reticulum. Why? Because if you take endoplasmic reticulum, you know Because of the electron microscope, we can study eukaryotic cell and we can identify the network or reticulum of tubular structure. So, endoplasmic reticulum is a tubular structure. It divides complete space inside the cell into two compartments. Inside the endoplasmic reticulum is intraluminal, and outside the endoplasmic reticulum is extraluminal or cytoplasm. In case of rough endoplasmic reticulum and smooth endoplasmic reticulum, there are two functions. Rough endoplasmic reticulum are involved in the protein synthesis and secretion. Protein synthesis and secretion, and uh, they are also in the touch with the outer membrane of the nucleus. Very important, outer membrane of the nucleus. Smooth endoplasmic reticulum, major site for the lipid synthesis, and in case of animal cell, lipid-like steroid hormone produced by them. Then, so detoxification is also given. I have seen somewhere, but you can eliminate also. Like Golgi body do not produce lipids. Endoplasmic reticulum SCR do. RER and mitochondria. By this you can eliminate. Sarah, incorrect statement about this figure.
These are round and oval cells.
Very good. Logically, it does not look round and oval shape. This is tracheid, and tracheid have thick cell wall, lignin cell wall, help in the transportation in the angiosperm, and it is dead and without protoplasm. Same diagram is given in the NCRT just for the different type of shape in the organism. See, this is tracheid, which is elongated, and this tracheid is dead, help in the transportation and present in the angiosperm. Correct. Next. Anvita. No, wait. Pratigya. I think I missed Pratigya. Pratigya. In animal cell, lipid-like steroid hormone are synthesized by in SCR.
Correct.
This is correct because we just finished this part. In smooth endoplasmic reticulum SER, major site of lipid synthesis, and in animal cell, lipid-like steroid hormone cholesterol are synthesized in SCR. You can write cholesterol. Add all the extra point cholesterol in SER. In SCR. SCR. Next, Pratigya. Convex or trans phase of the Golgi apparatus is mature phase from which lysosome arise. Pratigya.
True.
False. Because trans phase is known as concave phase, not convex. Everything else is correct. It is mature phase, and from mature phase, lysosome arise. Let's see in case of Golgi apparatus. In diagram, you have cistern, you have cis phase and trans phase. We'll discuss about this. Discovered by Camillo Golgi in 1898. First, observe as dense stain reticular structure. Dense stain reticular structure near the nucleus. Near nucleus, dense stain reticular structure, and consists of flat disc sac, also known as cisternae, and their size is 0.5 to 1 diameter, parallel to each other. Number vary in the Golgi complex. Cisternae are arranged near the nucleus, see, with distinct convex or cis or forming phase. Convex is cis phase, forming phase. Concave is trans or maturing phase. So this was the mistake. They gave convex trans. Correct. Remaining thing is easy. Okay. These two, cis and trans phase, is completely different from each other, but they are connected with each other. Their main function is packaging of the material, and this packaged material can be transported inside the cell or outside the cell. It depends upon the job. Whatever material is packaged, it will form the vesicle, and this vesicle came from the endoplasmic reticulum. When endoplasmic reticulum is associated with the cis phase of the Golgi apparatus, move toward the mature phase, and that's why Golgi body and endoplasmic reticulum are strongly associated. Why? You have a factory from which you produce, let's take protein. This is endoplasmic reticulum. Then you pack this protein in Golgi body packaging in a Golgi body, and after that you will get finished product, and this finished product is actually lysosome, which will do its work. So, okay, so all three should be associated uh, closely, so they can function smoothly. Number of protein synthesized by the ribosome in the endoplasmic reticulum is modified in the cistern of Golgi apparatus before they release from the transphase. And main function of the Golgi body is formation of glycoprotein and glycolipid. See, protein and lipid formation by endoplasmic reticulum. Glycosylation, glucose addition is by the Golgi body in the packaging process. So this is wrong. Remember, here it should be concave. Third, Patigya third.
True, sir.
Wrong, because if you see lysosome, we know four systems are present in endomembrane system. First, endoplasmic reticulum. Second, Golgi body. Third, lysosome. And fourth, vacuole. All are integrated, associated with each other. Come to this point. Membrane of lysosome have alkaline hydrogen pump which maintain alkaline pH. This is wrong. Acidic pH. Acidic pH. How do we know? See, next structure. Lysosome is a membrane-bound vesicular structure formed by the Golgi body after packaging process. If you take individual lysosome, it is rich in almost all type of hydrolytic enzyme, hydrolase enzyme. Example, all these are hydrolase enzyme, lipase, protease, carbohydrates, and these are active at acidic pH. Acidic, not alkaline, acidic. And because you have lipase enzyme, it will break lipid. You have protease enzyme, it will break protein. Carbohydrates will break carbohydrate. Nuclease, nucleic acid. So you can digest all these things inside our cell with the help of these enzymes present in the lysosome, and also known as suicidal bag. You are already aware about. They can eat the old cell. Autophagy is very popular job of this. And have you heard about we should have do fasting?
Yes,
So what happened during the fasting? If you do intermittent fasting 16 hour, 18 hour, so it will give enough time to the body if there is any dead cell present. So dead cell will be engulfed by this lysosome and we will feel young, energetic because dead matter is utilized. So you have only fresh active material. Next, Pratigya. D. Contractile vacuole of amoeba help in the osmoregulation and excretion. This is correct. How do we know? Last topic vacuole. Again membrane bound. Remember, all this endomembrane system are single membrane bound. All are one membrane bound. All four. Endoplasmic reticulum, Golgi body, lysosome, and vacuole. Correct? They have different function and structure, but they are considered as endomembrane system. Why? Because their function is coordinated, associated with each other. Correct? Since mitochondria, chloroplast, and peroxisome are not coordinated with the endomembrane system, they are not considered as a part of the endomembrane system. One thing. Here you can see nuclear outer membrane is associated with the rough endoplasmic reticulum and smooth endoplasmic reticulum is slightly away from the nucleus. Okay. Last vacuole. Again membrane bound, single membrane bound, contain waste material along with water, sap, excretory product, and other material which is not useful for the cell because it is like waste container, dustbin, kind of dustbin in the simplest term, dustbin of the body, but not in our body, plant body, because they don't have excretory system. Single membrane known as tonoplast. In case of plant, it can occupy, not always occupy 90%, but it can occupy 90% of the volume. In that situation, cell will die. What is the application of this tonoplast? Transportation of ion. But this is interesting. Against the concentration gradient means active transport. Means active transport. And during this active transport, concentration of the ion is high in the vacuole. So we need to invest energy. In case of amoeba, we have contractile vacuole for excretion. In other protista, food vacuole are also present that engulf the food. So, in amoeba, contractile vacuole are important for excretion.
so one doubt, sir.
Yes, Jordan.
So you mentioned that cell would die if it's at 90%, if the vacuum is 90%. Why is that, sir? Is it at 90% or above 90%?
No, even at 90%, because see, 90% if volume is covered, it means there is no space for the other organelle to act.
Huh.
And if there is no space for the other organelle, so they will be inefficient and ultimately die. What is the
Right. Okay,
purpose?
yes, sir.
So, what happen? You have a young plant cell that will have very
Hmm.
small um, vacuole size compared to its cell size. But as cell mature, there will be more waste material. Waste material will be stored in vacuole.
Huh. So the
So
more it
when
grows, more it becomes, and then it
exactly,
dies.
so when
Understood, sir.
90% it means cell is old, it will die, and ultimately when all the leaf cell die like in this manner, leaf will follow. Got it, Anvita?
Sir,
Anvita,
ribosome.
ribosome. Because mitochondria, Golgi body, and true nucleus are absent in case of bacterial cell. So, what is one structure that is present common to all the cell? Ribosome. So, this ribosome is crucial, and you get question from the ribosome. It is given in the prokaryote also, in the introduction part also, like here, introduction part here. So ribosome is non-membrane bound organelle. Non-membrane bound organelle are popular because you have mainly two, three. One ribosome, second centriole, third nucleolus. So these are the popular non-membrane bound structure. And it is written here present in all cell, whether you take prokaryote or eukaryote. Same question. Ribosome is from the same statement. Correct. Within the cell, ribosome not only found in the cytoplasm but also present within two organelle. It is present in the chloroplast. It is present in mitochondria. It is present on rough endoplasmic reticulum. Got it? Same ribosome is given here. Mm,
So they rang the bell for food. Can I go eat and come?
sure.
Thank you, sir.
No organelle like one in the eukaryote found in prokaryote except ribosome. See, this question asks from this statement. Correct. Third, they ask ribosome here. In prokaryote, ribosome is associated with the plasma membrane. Size is very small. Remember unit, nanometer, not micrometer. And made up of two units, 50 and 30, and forms 70. Protein formation. And when multiple ribosome attach to one mRNA, it is known as polyribosome or polysome. And what is the purpose? Again, protein formation from mRNA. Protein formation from the mRNA. And same ribosome is given here. Granular structure. First, observe under electron microscope because one of the smallest structure you cannot observe without electron microscope as dense particle by the George Palade in 1953. In the same year when we discovered DNA double helix model. DNA double helix model. This ribosome made up of RNA and protein and not surrounded by any membrane. Eukaryote ribosome is ATS, prokaryote 70S. S stand for Swedberg unit, which explain how much a material can sediment settle down. So indirectly it also measure is this material dense, like more dense or less dense, density. Sedimentation tell us density and size. Bigger size, more sedimentation. Correct? And both made up of two subunits. So, who is next? Who answered this part? Anvita. Marinder. Which of the following ensure genetic material variation in the meiosis?
So third option,
First we'll finish question from the ensure genetic variation in the meiosis. Crossing over. But we'll do it later. First we'll finish cell the unit of life question. Answer this question, Marinder. Depending on the membrane protein classified as integral or peripheral.
so fourth option,
Ease of extraction. Ease of extraction, how? Because as I mentioned, it's okay if you make mistake, we'll read about this. It is given in the plasma membrane, cell membrane, in eukaryotic cell. Detailed structure of the membrane is studied only after the advancement of electron microscope in 1950s because small structure. When we do chemical study of RBC chemical study, it tell us about the proper structure of the plasma membrane. If you study use electron microscope and study RBC, you get the proper structure of the plasma membrane. We know this plasma membrane made up of the lipid and this lipid arranged in the bilayer. Lipid arranged within the membrane have polar head on the outer side and hydrophobic non-polar tail on the inner side. What is the benefit of these two layer like polar head and hydrophobic tail? It makes sure that non-polar tail of saturated hydrocarbon means non-soluble part, water insoluble part is protected from the water. Water insoluble part is protected from the water. Here, lipid present in the membrane made up of phosphoglyceride means phosphorus is also present. Phospholipid it is. If you do chemical investigation, then you get to know ki this membrane made up of protein and carbohydrate also. And protein and lipid variation will be there between cell to cell. Like if you have one cell present in the skin, main purpose is protection. But if you have cell in the heart, main function is the contraction. So function is different. That's why percentage will vary. Example, if you take RBC of human being, this 52% of protein and 40% of lipid. 52% of protein and 40% of lipid. Now, this was the question, Marinder. Depending upon the age of extraction, depending upon the age of extraction, you can classify membrane protein into two parts. Integral means inside or peripheral means outside. Peripheral lie on the surface and integral is partially or totally buried in the membrane. Okay, as you can see here. You have cholesterol for the stability, sugar for the attachment, peripheral protein. This one is peripheral protein outside. Another one, you have integral protein integrated inside and lipid bilayer, two layer, one upper and another lower. Even one upper layer, this is lipid bilayer. So have two part, hydrophilic head and hydrophobic tail. Phobic tail. So two component are present. And this is the fluid mosaic model given by Singer and Nicholson in 1972. Year is also important. Improved model of the cell structure were proposed by Singer and Nicholson in 1972, known as fluid mosaic model. And according to this model, there is a quasi-fluid nature, like half fluid, half solid mix. Quasi-fluid nature of the lipid. Lipid have fluid nature as well as the solid part because protein are present. And this lipid, which is kind of liquid, is responsible for the lateral movement of the protein within the oral bilayer. I gave you example of gulab jamun in sugar water. So sugar water is like lipid, gulab jamun is like protein. Protein can move side by side. Gulab jamun can move side by side in this layer. And this quasi-fluid nature of the lipid have ability to move within the membrane. So you talk about the fluidity part. See, you know, cell membrane is soft and can interact easily with another cell. That's why there should be fluidity. If it's very hard and tough, what will happen? Nothing can enter inside the cell. Then it will be waste. Correct? That's why when you have fluid nature, then it will help to grow the cell because it is soft fluid. It can form junction with the other cell. It can secrete the material. It can eat. Endocytosis means eating, and it can help in the division. Everything is possible because of fluid nature, not hard. Make sense, guys? But today I will try to keep uh, discussion short after every question rather than teaching the whole topic. Next, this is from the interface. No, this is again from the cell cycle and division. First, we'll do cell metabolically active growth repair meiosis. Who is next? Chandana, 107.
Yes, sir.
Requires specific stain, vital stain to viewed under the microscope and involved in the oxidative phosphorylation.
It's in mitochondria.
Very good, mitochondria. Oxidative phosphorylation means, in simplest language, ATP formation and require vital stain for the observation. So let's do all this vacuole pattern mitochondria. Mitochondria, unless specifically stained, unless we don't stain it, we can't see under the microscope. One thing. Number vary from this cell, depend
So
upon the
what is the
activity.
stain which is used for mitochondria? Because I remember they asking something about the specific stain name for mitochondria. Some green. I remember some green stain it was.
Genus green, I think.
Oh yes, sir.
Genus green. There is very popular stain. I think something like genus green. Mitochondria stain.
Janus
Genus
green
green.
only, sir.
Yeah, genus green is very popular. So if you asked, you can write genus green stain to visualize the mitochondria. Second, uh, shape size. See, number is different. Even shape size is also different in different cells. Because as I mentioned, skin cells are less active, less number of mitochondria. Heart cell are more active, more number of mitochondria. Shape is variable, so sausage shape or cylindrical shape. Diameter can be variable, zero point 0.2 to 1, average 0.5, or length 1 to 4. You have to compare with uh, the cistern in the Golgi body. It was 0.5 to 1 in diameter. Cistern. Almost same range you have for the uh, diameter. Almost same range for the diameter because average is this, but length is more. You know most of the other thing, double membrane structure, outer membrane and inner membrane are present, and there are two aqueous environment, one between two membrane and second aqueous environment inside the inner membrane. It's like this is outer membrane, this one is inner membrane. You have aqueous water in this, and you also have aqueous compartment inside this. So two types, outer compartment and inner compartment. Outer compartment and inner compartment. Inside the inner compartment, you have matrix, liquid. So this part is known as matrix. Outer membrane form the continuous limiting boundary. Limiting boundary means simply boundary. Limiting boundary means it will limit the mitochondria. Inner membrane will form cristae because there is a folding like this. These folding are known as cristae. Cristae toward the matrix. So cristae toward the matrix is important. Then what is the role of cristae? Increase surface area. Each membrane of the mitochondria have its own enzyme associated with depend upon the function. Main function is aerobic respiration. You know respiration, electron transport system, oxidative phosphorylation, ATP formation. Correct? For ATP formation, we use electron transport system, ETF, electron transport system. And that's why it is known as powerhouse of cell, but you can also mention ATP formation. So oxidative phosphorylation. Oxidative means with the help of oxidation process, This is phosphorylation, addition of phosphorus and formation of ATP. What are the things present in the mitochondria? We have, like bacteria, single circular DNA, like bacteria, single circular DNA, RNA, ribosome 70S type, protein component required for the protein synthesis, and divide by the division. Everything look like bacteria. Most of the component inside the mitochondria look like bacteria. So we assume that during the evolution, bacteria converted into the eukaryotic cell and bacteria form mitochondria. That's why semi-autonomous organ. This word is also important. We are using longitudinal section of the mitochondria because majority of the structure you see transverse section. Here we have longitudinal. Like what I showed you, this is the transverse section. But what shown by the NCRT is longitudinal section. This is what you learn in this class, where you need to focus on. The star metrics are important. Space between the two membrane is intermembrane space. This one between two membrane, inner membrane, outer membrane. So this is done. Pectin, zygotin, Jordan. Two for the centriole. And
Centrio.
also, I know it can be boring for you if you don't write anything. You have to write. It is compulsory. I will check your notes.
Yes. So one is true. Non-membrane bound cell organelle helps in cell division. True.
Mm.
Their core is axonium. Uh, true. No, it's not.
In cilia flagella, you have axoneme.
Okay, it's not a centriole. Uh, 9 microtubule doublets at periphery. It is true.
Triplet.
Doublet. Oh,
You
I'm
have
confused with
triplet in
that.
centriole. Doublet are present in case of
Cilia
cilia flagella.
flagella. Okay.
Mm.
Cartel organization is present. That is true. And then a 9 plus 0. No. 9 evenly spaced is there. That's what they mentioned. So 9 plus 0 is just 9. Correct.
Because 9 plus 2 is present in cilia flagella, 2 are central. Here you don't have any central thing.
Have the center. Yes. So it's 9
So
plus
three
0. So it's
statement,
even.
yes. So
It's
three
only
statement,
2. 3 statements are correct. So it's
got it?
2. Option
So
2.
you can
Yes, sir.
write, we are done with the centriole. Cartwheel organization in each centriole we have seen. Non-membrane bound cell organelle in centriole we have seen. 9 plus 0 organization, 9 uh, peripheral microtubule triplet are present, no central. So you can write here 9 plus 0 arrangement. 9 plus 0 organization. Because there was statement given in the from the cilia flagella, you can do this. Cilia flagella, hair-like outgrowth from the cell membrane. Now you are talking about cilia. Remember, you are talking about cilia. Small structure work like ore. Ore, we use in the boat. These plastic structure that help in the movement of water. That is ore. And cilia are responsible for movement of either cell or surrounding fluid. Come to the flagella. Flagella are longer and responsible for the cell movement. Prokaryote bacteria is also present, but prokaryotic bacteria is structurally different from the eukaryotic flagella. Okay. If you study the electron microscope, cilia flagella, they are covered with the plasma membrane, so membrane bound structure. Membrane bound structure. But centriole is not membrane bound. Centriole is not membrane. Okay. Its core is known as axoneme. The core is known as axoneme in cilia flagella. रुक जाए, रुक जा। Poses microtubule are parallel to each other and parallel to the long axis. Axonym, if you see, have 9 pair of doublet. See, in case of central, you have 9 pair of triplet. Like 9 triplet, not pair. Here you have 9 pair of doublet. Radially arranged peripheral microtubule. And one extra at the center. This arrangement of the axonym microtubule, within the axonym, you have this arrangement. Such arrangement is known as 9 plus 2 arrangement. 9 plus 2 arrangement. 9 outside, 2 center. Central are attached to each other with the help of a bridge and enclosed in a sheath. I'll show you the diagram. NCERT have given this diagram. Connected to outer side doublet with the help of the radial spoke. So there are total 9 radial spoke. And peripheral doublet are also connected with each other with the help of linker. Both cilia and flagella emerge from the centriole-like structure known as basal body. Look at this. Here you have transverse section. You have outside peripheral microtubule doublet covered with plasma membrane. This question can be asked and you will be confused in the plasma membrane. Let me tell you. You will be confused in the plasma membrane because you will feel, oh, centriole do not have membrane. So this cilia flagella will not have plasma membrane. No, it have plasma membrane. Two microtubule doublet are connected with each other with the help of inter-doublet bridge or linker. Okay, you have central microtubule, but here you don't have doublet. These are separated by the bridge. Separated by the bridge. You each microtubule doublet have one radial spoke, so total 9 radial spoke. Total 9 radial spoke. And the covering of the center is known as central sheath. Central sheath is attached to the peripheral doublet with the help of radial spoke. Okay, so this core that contain all these things, this core is known as axonym. This core is known as axonym. Right? And here you should know what is this. This is electron microscope structure of the cilia in flagella. Electron micrograph. And here internal. Yeah, got it. So this part is done. Cartwheel shape structure. Tuhina, are you there? Tuhina? No. Sarah? Microtubules are made up of Sarah. Tubule protein. How do we know? In centriole, we just finished. Key centriole made up of microtubule. Remember, we said peripheral microfibril. Na? So here in this centriole, you have microtubules. And as the name suggests, in microtubule, there is tubulin protein present. Tubulin protein present here. Both in whether you take cilia flagella or centriole, you have tubulin protein. Next. Wow, we got same question. Pratigya. So no need to teach this part. Pratigya. First statement. Wrong. Because you have 9 peripheral microtubule doublet, not triplet in cilia fibula. Second, Pratigya.
Not correct.
Cilia are smaller, which work
Third
like
one also
hole.
correct.
Correct. Fourth,
That is plasma membrane, not cell membrane. D1 is incorrect. Plasma membrane, na?
plasma membrane, cell membrane, same thing. The cell membrane or for the cell plasma membrane outer covering. But overall meaning is membrane.
No.
Got it?
Yes.
And cell membrane, plasma membrane, same thing. Only thing is plasma membrane or cell membrane made up of exactly same thing. Most of time plasma membrane is generalized word, cell membrane for cell, but thing are exactly same. Next, Anvita, first statement.
True.
Yes, crystal increase surface area in case of mitochondria in the membrane. Correct. Next,
Second one is also true.
thylakoid you have correct. Next,
Third one is wrong, no? I
no,
think.
mitochondria matrix have Krebs cycle, na?
Oh,
Respiration,
yeah.
respiration in plant.
Hmm.
And fourth one,
Fourth one.
what is the right statement instead of stroma? Cytoplasm,
Yeah.
cell cytoplasm, caryophysis. Correct. So you can write here also. Where we wrote this diagram of matrix. In this matrix, you have Krebs cycle, happens citric acid cycle, Krebs cycle will happen. If you see plastid because thylakoid was given, so we can finish in between. Present in all the plant and euglenoid because all these make food. Easily observed under the microscope because of the large size. Have some pigment in color and that's why it has specific color in the plant. If green color chloroplast, any other color chromoplast, no color leucoplast. First is chloroplast with green color. Why green color? Because of the chlorophyll and carotenoid pigment. And it is responsible for trapping of the light energy and essential for the UV. No, we have full photosynthesis receptor based on this. Second number is chromoplast, very popular fat soluble carotenoid pigment. Fat soluble carotenoid pigment, carotene, xanthophyll, very important.
I'll just be back in 5 minutes.
Sure. So and have different color. It could be yellow, orange, red, like orange color, red color in the cherry, yellow color in some other fruit. So all this is responsible for the chromoplast, like red color in the tomato. And third is leucoplast, colorless, because there is no pigment as such. That's why colorless. And shape and size will be different. Main role is the storing the nutrient. So leucoplasts are further different type. Example, it can be amyloplast, carbohydrate storage. Example in case of potato, amyloplast. Elioplast, oil and fat, and aleuroplast, protein, aleuron layer, protein layer. Amylose, so potato. Majority of chloroplasts are found in the mesophyll cell because mesophyll cell in the leaves are responsible for photosynthesis. Shape can be different, lens, oval, spherical, disc, ribbon shape. Ribbon shape is something you might miss. Other you can still get idea. Oval, spherical, disc, shape, lens. Size is very big. 5 to 10 micrometer in length and width is this. Way much bigger than the mitochondria. And number can also vary. In case of Chlamydomonas green algae, you have one chloroplast in one cell, but you have 20 to 40 chloroplast in mesophyll cell. So number of chloroplast in mesophyll cell are two. If you have one cone-like structure, thylakoid, and group of cone-like structure is grana. Liquid in case of chloroplast stroma, liquid in case of mitochondria is matrix, and two grana are attached with each other with the help of stroma lamella. Again, double membrane structure, so outer and inner membrane. But this was 2024 NEET question, I guess. Inner chloroplast is less permeable logically because outer membrane can allow things easily, but not inner membrane. Space inside the chloroplast is known as stroma, and you have flat sac known as thylakoid, and all this thylakoid is present in stroma. Thylakoid is a like pile up of coin and grana. But more important is grana is also known as intergranal thylakoid. Grana is also known as intergranal thylakoid. You have flat membranous tube. Remember, flat membranous sac is thylakoid. Flat membrane tube is stroma lamella that connect thylakoid of the different grana. Thylakoid have a space known as lumen, space within the thylakoid. And stroma of the chloroplast have enzyme required for the photosynthesis, carbohydrate and protein. Remaining thing is exactly like mitochondria. Single circular DNA, ribosome, chlorophyll pigment are present in thylakoid for photosynthesis, and ribosome is 70S type. Look at this. Thylakoid light reaction, photosynthesis. Krebs cycle matrix, cistern this, glycolysis, cytoplasm. Stroma have dark reaction. Stroma have dark reaction during photosynthesis. Who answered this question? Pratigya. Anvita, are you there? Anvita. Marinder.
Yes, sir.
Sir, I'm back.
Okay, to Hina.
So third option.
Third option. So, question is asked about the cytoskeleton. I will teach cytoskeleton and then you tell me what is the answer. These cytoskeleton is the big network of the protein structure and present in the cytoplasm. Cytoskeleton are involved in the mechanical support, motility, and shape maintenance. That's all. And this cytoskeleton involve this microtubules. What is this? Microtubule, microfilament. Filament means long rod. Tubule means wide rod. And intermediate. These are the three structures present in the cytoskeleton. Made up of mostly actin protein. Filament na actin protein. Now, Marinder, tell me answer of this question. Filamentous or tubular because microtubules are present, right?
Yes,
Yes, sir.
And second, mechanical support and motility, mostly made up of the actin protein, not tubulin. Tubulin, microtubule in case of centriole, flagellin in flagella. Flagellin in flagella. These are not flagella. Sandana. Vandana.
Sir, I'm not sure.
Option 1,
9
no,
plus
sir.
2, 9 plus 2 arrangement, na? In case of cilia and flagella. The radial spoke is 9. Yes, Jordan, you are asking something?
So it was option 1, no, sir.
Yeah. See, if you see here, you have 9 plus 2 arrangement, where 9 pair of doublet, 9 radial spoke, correct? And 2, a pair of central located microtubule. Correct? So 9 plus 2, 9 radial spoke, 9 doublet, and 2 center. So you can write here. Here you have 2, you have 9, you have 9. Got it, Channa?
Yes, sir.
Jordan.
Incorrect answer. Central, page central, cognition, country. Uh, Tubulin protein, no, sir. Option 2.
First option, correct?
First option is correct, yes, sir.
Second,
Second is false, sir, because it says flagellin protein and not tubulin.
tubulin protein, not flagellin.
Yes, sir.
Third,
And each central has cartwheel arrangement organization, correct. Centrals usually contains 2 cylindrical centers, correct.
yes. Any doubt?
No, sir.
Tuhina.
Cell membrane, sir.
Yes. Cell membrane of the prokaryotic and eukaryotic look similar. This is given in the introduction part. Yes. Plasma membrane is semi-permeable in nature, interact with the outside world, and this membrane is structurally similar to the eukaryote. So prokaryote and eukaryote plasma membrane is same. Correct? Uh, Tara. What is the shape of RBC? Columnar epithelium cell, nerve cell,
The nerve cell.
is branched and long,
Yes, sir.
mesophyll and round and oval. All the diagram given in the NCRT. Next to this. See. First RBC round and biconcave. WBC amoeboid irregular shape. Columnar epithelium long and narrow. Nerve cell branched and long. Trachea elongated and mesophyll round and oval. Even you can correlate if you have seen the diagram of all these. Next. Anvita, are you there? She is not.
Yes, sir. Yes, sir.
Okay. Then answer this question. 120.
First one is correct.
Hmm.
1 second. Second also is correct.
Hmm.
So it's
Third,
correct only, no?
they do not explain how new cell arise.
Oh, wait, yeah. Okay. That was better.
That was virtue, yeah.
Centriole is a single membranous cell organization. Is this correct?
No, ma'am,
Oh,
not
wait, that's
membrane.
wrong. Fourth one is correct. So,
So 1 and 4 is correct. First is physiochemical approach. We have studied this. I will do it again here. Biology is the study of the living organism. Detailed description of the form appearance, bring the diversity. Cell theory explain this. A cellular organization is present in the all living form. Description of the cell, how cell divides is important in this chapter. Cell theory that explain the physiological behavior, physiological aspect and behavioral aspect, how cell interact with each other and they integrate, and there is a physiochemical approach, and we use cell-free system to explain the physiological and behavioral process in the living cell. If you want to study living cell, how living cell work, what is the behavior, you have to study the structure and chemical present in it, and then you have to um, do the experiment without the cell. All these things explain us about the molecular form. See, there are two forms. One is the as a organism you study the life form. Second, inside the cell you study the chemical. So here we are talking about inside the cell molecular term. So further you can analyze based on the this element and compound present within the cell, which organic compound is present, and everything that you have in the biomolecule chapter in the same unit. Correct? Which compound, which chemical, which organic compound, everything. Protein, carbohydrate, fat, lipid. So this explain if there is any abnormal process occur in when we are not healthy, like when we are ill, there must be some change in the chemical composition. We want to study that. And for this, we use physical or chemical approach to study and understand the living form. This is known as reductionist biology. Correct? One thing. We already did this, I guess. Ramachandran studied from the Madras School of Conformation Analysis of Biopolymer. Analysis of biopolymer like protein, polysaccharide, nucleic acid. He discovered triple helical structure of collagen in Nature book 1954, and he used Ramachandran plot to understand the biology. Later on, nothing else is important. This Linus Pauling will explain the alpha helix and beta plated sheath that help him to understand the collagen, because collagen is protein made up of alpha and beta helix sheath. Correct. Come to our point. Skeleton and Swan formulated cell theory, but this theory did not explain how new cell arise. There was a new guy known as Rudolf Virchow in 1855 who first time explained that how old cell divide and produce new cell, omnicellula, ecellula, pre-existing cell, omnicellula, ecellula. So he modified the hypothesis of Skeleton and Swan. They gave hypothesis and gave the final shape. He all living cell, this and new cell arise from the old cell. So it is the combination of three: Skeleton, Swan, Virtue. So this statement done. I think we left with this is done. Prokaryotic lac membrane monoclonal is given in the next paragraph. though it is basic. Hmm. Cell of the human chick, animal cell, have outer boundary membrane delimiting structure of the cell, because cell membrane or plasma membrane is the delimiting structure. Inside you have nucleus. Nucleus contain chromosome. Chromosome contain DNA. Cell have all membrane bound structure is eukaryotic, and cell lack membrane bound structure in case of prokaryote. Both have semi-fluid matrix cytoplasm, and cytoplasm is the main arena of cellular activity in both plant and animal. And chemical reaction happen in case of cytoplasm will keep it alive in the living state. Then, next after Barinder, what is A, Barinder? B,
It's a bridge.
C,
Microtubules.
central one, and D, radial spoke. Total 9.
Spoke.
Correct. So A is plasma membrane. Interdoublet bridge. B is correct. C, central microtubule. D, radial spoke. Sandana,
Yes.
peroxisome. I will help you in this. In photorespiration, you utilize peroxisome. And same peroxisome are this body. Microbodies include peroxisome involved in photorespiration, lack in C4 plant. Next, Sandana, Christy. Next, Sandana.
It's a nucleic acid.
RNA is nucleic acid. Next, vacuole. Vacuole covering is chromoplast.
Yes. Sister is
Disc-shaped structure present in the Golgi body. Jordan.
A researcher observes maximum ATP production in cells with highly folded mitochondrial membrane. This is because matrix volume is increased. Option 3, sir.
Logically, because there are enzymes available for the ATP synthesis, and these enzymes are present in the crystal.
In the crystal, but I mean, if the surface exit, like more surface area shouldn't it be that way,
But
sir?
even if you increase the volume, but you don't have enzyme, what is the use of increasing volume?
Okay, that would make sense, yes, sir.
Vina,
Um, next, protein synthesis, sir.
protein synthesis, because synthesis is the main function of RER.
Yes, sir.
So you have to write in SCR, there will be detoxification, there will be lipid synthesis and calcium storage. I think I have seen in summary. I have to check
Yes, sir,
summary.
there was another question.
Okay, I have seen this, like I have taught this, so I will write here. Doesn't matter. I will write in SCR, and you have to also mention the same thing. SCR, SER have detoxification. This is given in NCRT, I think in summary, in new summary, and calcium storage. Wajiba. Got it? So from this paper, many things from this chapter is done. Majority of part of the chapter, but we haven't seen many questions from the not many, not even a single question from the nucleus part. So we need to do more. And cell wall part, transportation part, there was no questions. And there was no question from mesosome. Mesosome is important. So 70-80% part of the part is done in just 1.5 hours, less than 1.5 hours. Okay. So uh, take a break of half an hour, and then we'll continue with the cell cycle and cell division question. And then again we'll come to the uh, cell the unit of life after doing cell cycle and division question. Got it? All of you need break?
Yes, sir, for some time.
Yeah, half an hour is, I guess, more than enough. So this is important feature of prophase. Key centriole, which undergone duplication in S phase, centriole will now begin to move towards the opposite pole. And here, one thing is chromosome material will condense, and it will form compact mitotic chromosome, mitosis chromosome. And one chromosome have two chromatid attached together at centromere. How? Example, this is one centromere. Here you have one complete chromosome. 1, 2, made up of two chromatids. This is one, this is second. Okay. And second, initiation of assembly of mitotic spindle will start assembling here. Microtubule will assembling here. Protein component of this cell will assembling here in the process. And you will have ester formation, star-like structure. 2, star-like structure. Microtubule arise from centriole. From centriole. So centriole plus this star-like fiber-like structure like this is known as ester. This is not written here, but in earlier NCERT you used to get. Last, at the end of prophase, if you see under microscope, you don't get all these structure: endoplasmic reticulum, Golgi body, nucleolus, nuclear envelope. Why? Because your DNA amount is double. This double DNA amount will put pressure on the wall and wall will break. If you break the nuclear envelope, Golgi body and endoplasmic reticulum will also disappear because they are associated with the cell membrane. And if cell membrane, nuclear membrane, if nucleolus wall break down, nucleolus will also not visible. So this is early prophase, you have less compact material, and late prophase, you have more compact material with broken wall, nuclear envelope. So you should know what is the characteristic feature of each. If I have to summarize into 2-3 word, main feature of prophase is condensation of uh, chromosome material and movement of centriole toward the opposite pole. Movement of centriole toward the opposite pole. That's all. Other than that, important point key, at the end of the prophase, many structure will disappear. And this is the transition to the metaphase. So it is lie between the prophase and metaphase. Why? You have proper condensed chromosome. You know, each chromosome made up of two uh, each chromosome made up of two chromatids. Okay, here you have centriole with fiber. This is known as ester. Okay, and what happen in case of metaphase? All will lie on the equatorial plate, and these are the spindle fiber. Spindle fiber will attach to the kinetochore. Spindle fiber will attach to the kinetochore. These are the characteristic feature of metaphase, and chromosome is clearly visible. See, keyword, complete disintegration of the nuclear envelope will start the second phase. The second, chromosome will spread throughout the cell. Third, chromosome condensation will be complete. That's why they are clearly visible. This is the stage where morphology of the chromosome can be easily studied. You can study chromosome. Why? Because chromosomes are clearly visible here. Each chromosome made up of two sister chromatids and held by the centromere. You have a small disc-shaped structure at the surface of centromere. This structure is known as kinetochore. And at this kinetochore, spindle fiber will attach. And it will help to move the position of the chromosome. You know, it will move, they will hold this up. Chromosome will lie at the equator. Okay, where spindle fiber of one centromere will attach to the kinetochore. And same from the another pole. So this plane of alignment of chromosome is known as metaphase plate. So these are the characteristic features. First, spindle fiber attached to the kinetochore. And second, chromosome move to the equator and align at the metaphase plate. So what are the key features of metaphase? One is alignment of chromosome at metaphase plate at equator. Second, chromosomes are clearly visible in this state, made up of two chromatids. And third, spindle fiber from one centriole are attached to the kinetochore on one side. Remember, one chromosome have one chromosome have two kinetochore. That's why both side. If you have 46 chromosome in one cell, so 92 kinetochore will be present there. Third, anaphase. This is responsible for duplication of chromosome. So remember, S phase is responsible for DNA duplication, and anaphase is responsible for chromosome duplication. Very, very, very important. This point. You see change only in these two states, S and anaphase. S for DNA replication and anaphase for the chromosome. Okay, next. If we see anaphase, each chromosome will split simultaneously, and two daughter chromatid will be known as chromosome of the future daughter. As you can see, chromosome number double. Chromosome number double. Migrate toward the opposite pole, toward one pole. That's why you will have leading edge and one trailing structure backside. So you can see here, one leading this, one part is lead, and another part is backside trail. This lead, this trail. Okay, so anaphase. So these are the characteristic features. Centromere split and chromatid separate, one thing, and chromatid move toward the opposite poles. Chromatid move toward the opposite poles. Last is telophase, exactly opposite to prophase. Everything. Example: chromosome decondense. In prophase, there is condensation of genetic material. Here, decondensation. Lose individuality. Their chromosomes start become individual alone. Individual chromosome no longer seen. There you see individual chromosome. Chromatid material collect in a mass at the two pole. There in one chromosome, as you can see here. Individual chromosome in prophase. So chromosome lose their individual entity. Nuclear envelope will assemble around the chromosome, and all the structure that disappeared during the prophase, whether it is Golgi body, endoplasmic reticulum, nucleolus, and nuclear envelope, all will reappear here. Exactly opposite. That's why we said. And last point, cytokinesis. Remember, it is shown here that this is telophase. Okay, this is anaphase. Here, this is the stage after cytokinesis, not interphase. This is wrong. This should be after cytokinesis. One cell become two. One cell become two. Okay, interphase is wrong. This is wrong given in the NCRT. So, mitosis is not only karyokinesis, but it also includes cytokinesis. In case of animal cell, there will be formation of the furrow in the plasma membrane. Furrow will deepen with the time and join the center and divide into two. In case of animal cell, if you take plant cell have cell wall, inextensible cell wall, like you cannot break. That's why mechanism is different. Here, uh, cell wall formation start at the center of the cell. See, in case of animal, it start from the outside. In plant, it start from the center, and it grow outward to meeting the lateral wall. Outward. So it is kind of centrifugal. It is kind of centrifugal. And this is go deepen toward the center. So this is kind of centripetal toward the center. Okay. And this cell wall formation begin from the simple precursor known as cell plate formation. And this cell plate formation represent the middle lamella between the cell plate ultimately convert into middle lamella that will separate two plants. Cytokinesis, same thing. During the cell division, mitochondria, plastid, all the structure will distribute between the two cells. Sometime there is multiple nucleus, karyokinesis is done, but there is no cytokinesis. It can lead to the multinucleated condition and lead to the syncytium formation, multiple nucleated condition. Syncytium means multinucleated condition, multiple nucleus in a single cell, and that happen in liquid endosperm. Multinucleated condition. multiple nucleus in single cell. Because karyokinesis is not followed by cytoplasm. There is keep on karyokinesis. How? You had one cell with one nucleus. Karyokinesis occur and this cell will have two nuclei. But again, it is not followed by cytokinesis and karyokinesis again occur. What will happen? Four nucleus. So what is all this stuff? Multinucleated condition. Now we can solve many questions, I guess, using the same. Whose turn is this? Tuhina. Are you there? Tuhina. She must be reading. Tara. Cell contain 20 chromosomes at metaphase. Will have how many chromosomes in each daughter cell? Tara.
20.
Very good. 20. Because this is the cell have 20 chromosomes at metaphase. Then in anaphase, chromosome number will become double, 40. And after cytokinesis, cell will separate and each cell will have 20 chromosomes. So you should know in S phase, DNA replication. In anaphase, chromosome replication. Pratigya, correct statement. First, Pratigya. Barinder, 104. Barinder, are you there? Barinder?
Yes, sir. So first one is intranet.
Hmm.
Second is also intranet.
Third,
Fourth is intranet, sir. Then third is tranet.
checkpoint are the point that basically cross check is everything fine? Is there any mistake? What is happening in the cell point? So there are different checkpoints before S phase, after G2 phase, one during the meta phase. So there are different checkpoints to check whether everything is perfect or not. Next, Jordan. No, 106.
So can you hear me?
Yes.
So 105, 106. Okay, huh. The term equational division matches to meiosis 1, meiosis 2, binary fission, no. Amitosis, no. Equational division, won't it be option 1, sir? No, not option 1. Meiosis 1, that is no. So that will be 4, 2, 2. Option 2 only, sir.
Meiosis 1 is reductional division.
Reductional
Chromosome
division, and the
number will
meiosis 2
become.
is like proper is equational.
It is like mitosis. That's why equational
Yes,
division.
sir.
Sir, I have a doubt in the previous question.
Yeah. Ask your question.
I also marked meiosis 2 only when I was doing the paper, but then binary fission, isn't that also equational division?
No, because there we are not talking about the chromosome number, remains.
It's just multiplication there.
Yes, it is simply
Okay, sir.
multiplication.
So what about
So
amitosis?
amitosis is like it happened in the prokaryote,
Yes, sir.
where it is simply like multiplication, like binary fission or multiple fission, but proper mitosis, proper arrangement of the chromosome material is not there. It is simply
Okay, sir.
like one split into two.
Yes, sir, understood.
So, hmm. Tuhina 108.
Yes, sir. Um, sir, I can't. Okay.
First one,
That's correct, sir.
second one,
I think that's wrong. I think that's wrong, sir.
okay. Third one,
Correct, sir.
fourth one.
Correct.
So in metaphase 1, you have two equatorial plates because double number of chromosome.
Sir, but when I was doing the paper, they said second one is wrong. I also marked the third one because I thought two equatorial plates, but then.
No, there is a tendency of recombined homologous chromosome of the bivalent to separate each other during the diplotin stage.
So that's the thing. I marked the third option when I was doing the paper, but then they said that was wrong. I think it might have been a mistake from their end.
Diplotin stage, tendency of the recombined homologous chromosome of the bivalent to separate from each other. Correct. So this is correct.
Yes, sir.
Yeah. Uh, Here we can talk about meiosis because there will be more questions from meiosis. And meiosis, for meiosis, you should know mitosis. That's why we did mitosis first. Now come to this topic. Gametes are produced by meiosis process, and there will be half number of chromosome at the end of the meiosis process. Correct. The specialized diploid cell will produce this gamete meiocyte. These are known as meiocyte cell that undergo meiosis. Correct. Chromosome number will become half and processes meiosis. Fertilization will restore the diploid because two gametes fuse uh, during the fertilization process and it will restore the diploid phase. Meiosis will ensure the haploid phase of the life cycle. It occurs both in the plant as well as animal. Occur in the two cycle, but DNA replication occur only once. Example, if you have 2C amount in normal cell, diploid cell, and if it undergo one DNA replication, it will become 4C. Then this 4C divide into 4 equal cell. C, 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 C. Done. Okay. Next, meiosis one is initiated after parental chromosome have replicated to produce identical sister chromosome. Basically, yes, DNA replication. It involves pairing of homologous chromosome and recombination. These are the two important features. Sarah, what do you mean by homologous chromosome? Sarah, what do you mean by homologous chromosome?
Homologous chromosomes. I don't know, sir.
It's okay. Tohina, what is homologous chromosome?
Sir, the ones that are present on the, like, I don't know how to explain it, sir. Like
Jordan?
in a tetrad, it'll be two of them.
Okay. Jordan?
So can you repeat the question again, sir?
What is homologous chromosome?
So homologous chromosome is like when the two sister chromatids are the same, no, sir, from the same parental cell. There's no crossing over, so they're the same.
There will be crossing over in the homologous chromosome. NCRT mentioned this. Sandana, what is homologous chromosome? Answer is, example, our body have 46 chromosomes.
So and
Out
like
of
each loci will have the same thing, no, sir. I think it's that the genes are the same.
46 chromosomes, 23 chromosomes come from mother and 23 chromosomes come from father. Okay, 23 chromosomes come from mother and 23 chromosomes from father. It means, example, if we have one set each in every case, here you have one set, here you have one set. So if you see one set of each, what will happen? Chromosome number 12 of mother will look exactly like chromosome number 12 of father. There is small difference, but look wise more or less it is same. That is known as homologous chromosome. Correct? These chromosome number of 12 from mother and chromosome number 12 from the father will be the homologous chromosome. Got it? That's why you are talking about diploid and haploid, not triploid, because both look similar and you will see exchange between these two chromosomes. Okay? Chromosome number 12 of mother, chromosome number 12 of father, because they have different origin when they mix with each other, so produce new gamete. It produce new gamete. Homologous chromosome is that. Okay. Come to the meiosis one. Most important part of this chapter. Very long and complex than the normal process. That's why we divide into 5 further categories. First one, lactotin. Chromosome in this case gradually visible under light microscope. Okay, under light microscope. And second, condensation, normal thing. Second stage, zygotin, very popular. First thing, during this zygotin stage, both chromosome pair like uh, homologous chromosome, like chromosome number 12 from mother, chromosome number 12 from father will come close to each other and they will mix with each other. That process is known as synapsis. Process is known as synapsis. and the chromosomes that are being paired is known as homologous chromosome. Chromosome that being prepared is homologous chromosome. And if you see in the electron microscope, then this chromo synapse chromosome have a complex structure. This complex structure is known as synaptonemal complex. This structure is known as synaptonemal complex. So process is synapses, result, final product is synaptonemal complex. This synaptonemal complex, because it made up of two chromosomes, chromosome 12 of mother and father, this is known as bivalent. Bi means two, valent means for this topic chromosome, two chromosomes. And tetrad means four chromatid. Okay, so bivalent means two chromosome and tetrad means four chromatid. And bivalent tetrad or synaptonemal complex are exactly same thing because synaptonemal complex made up of two chromosome and four chromatid. Okay, but all these synaptonemal complex, bivalent or tetrad, is more clearly visible in the next stage, which is Pachytene stage. You never forget this. This is most popular step. Now there will be further condensation. Bivalent will be clearly visible here as tetrad. Why? Because all the four chromatid are clearly visible. Next thing, appearance of recombination nodule. Recombination nodule means when two chromosomes are mixing with each other, it form nodule types, a mass structure. That is recombination nodule. At the place of recombination nodule, we have crossing over, mixing of genetic material between non-sister chromatid of homologous chromosome. What is the meaning of sister and non-sister chromatid? You have a chromosome. This is centromere. This one is chromosome with two chromatid. This is chromatid 1. This one is chromatid 2. Chromatid 1, 4 chromatid 2 is sister chromatid. These are sister chromatid. Same way, you take another chromosome. This is chromatid number 3. We are just labeling number. Otherwise, this chromosome also have only two chromatid. Okay. So 3 and 4 are sister chromatid to each other because from the same pair. But chromatid number 3 and 2 will be non-sister chromatid. Same way 1 and 4 will be non-sister chromatid. Correct? 2 and 4 will be non-sister chromatid. 1 and 3 will be non-sister chromatid. From other, it is non-sister. From same chromosome, sister. Any doubt, guys? Anybody have doubt in this? What is the difference
Also,
between sister and non-sister chromatid? So during this crossing over that is happening between non-sister chromatid and mixing of genetic material, there will be exchange of genetic material between the homologous chromosome and non-sister chromosome. Enzyme involved in this process is recombinase because recombination process, and due to this crossing over, there will be recombination of genetic material in the two chromosomes. Basically, this recombination of genetic material between the two chromosomes is responsible for genetic variation. We are different from our sibling and uh, from our parents. Reason is this crossing over process only. Reason is this recombination process only. So recombination will be completed by the end of Pachytene stage, and chromosomes are still linked at the site of crossing over. In diplotin stage, in the starting beginning, now this synaptonemal complex will start dissolving. Like it will start, chromosome will start moving away from each other because there is already maximum mixing. Dissolution of the synaptonemal complex. Chromosome will start moving away from each other except crossing over site, there is a complete separation of the chromosome. And there will be X-shaped structure formation. This X-shaped structure is known as chiasmata. Chiasmata means X-shaped structure. In case of some vertebrate, this diplotin stage can last for month or year. I'll explain this point properly. Just give me a minute. So, guys, what happened in this case? Females start producing, like, start, not, I'm not saying complete process. Female start producing secondary, not secondary oocyte, oocyte when she is there in the mother womb. But actually, she produce secondary oocyte or egg after puberty. So there is a gap of 12-13 years when female is not producing any oocyte. This During this time, this oocyte will remain in the diplotin stage. Understood? In human being, oocyte almost for 12-13 years will remain in the diplotin stage because female start producing the process but not completing until 12-13 years. After that, there will be menstrual cycle and regular release of the secondary oocyte. Understood, guys? Anybody have doubt in diplotin stage? And last final stage, dikinesis, where last thing, this X-shaped structure will also end terminalization of chiasmata. This X-shaped structure will also end terminalization of chiasmata. What will happen? Chromosome, same normal process of prophase, condensation of the chromosome completely. Meiotic spindle will assemble to prepare homologous chromosome for separation. So meiotic meiotic spindle will assemble. By the end, same thing, nucleolus appear, nuclear envelope broken down, and this is the transition to the metaphase. Because after that, metaphase will start. Done. So what happened? Only thing is, other than the normal prophase, we are dividing further into 5 categories because it is more complex. First, chromosome come close to each other in the process of in zygotic stage. In pachytene stage, this closed chromosome will exchange their genetic material. Pachytene stage start moving away from each other. Diplotin stage, and ultimately, X-shaped structure will, like, they will completely separate. Diagnosis done. Why this unique step for variation? Tell me, guys. Anybody have doubt? if you see this other stage are exactly same as normal mitosis except anaphase 1. Example, in metaphase 1, bivalent chromosome will align at equatorial plate. Same microtubule will come to the opposite end. Everything is ditto same. Tuhina, are you there? Tuhina? Guys, can you see
So
two
did you
metaphase?
call me?
Yes.
Sir, I'm
Can
doing
you see
question.
two
Sorry.
metaphase plate here?
Yes, sir.
Sir? And guys, other than that, you can see this is kind of, you have this chiasmata stage, diplotin stage, where two chromosomes mix with each other. Here you have two chromosomes. Anaphase 1 is interesting because here no splitting of chromosome. No splitting of chromosome. Why? Because you have complete chromosome. So complete chromosome will separate. Sister chromatid will remain associated. But if you compare with the normal mitosis, look at this. Centromere will split and chromatid separate. This step, which happen in normal anaphase, does not happen in case of anaphase 1. Here, instead of chromatid, chromosome will move toward the opposite pole in case of anaphase 1. Everybody understood? Because you have pair of chromosome, homologous synapse, bivalent tetrad. Everything else is more or less same. Nuclear envelope nucleolus will reappear in telophase, cytokinesis, and dyad of cell. Dyad of cell means two cell. It's just after meiosis 1, your 2n cell will divide in n condition. Although in many cases chromosome do undergo some dispersion, some uh, what do you say, relaxation, but they do not reach to the extremely extended state. Instantly they will go to the next stage, that is meiosis 2. That's why between meiosis 1 and meiosis 2 you have very short gap, that is known as interkinesis. Interkinesis, which is very shortly. You don't have proper interphase like normal mitosis. It is very short phase because after that you have meiosis 2. And each and everything of meiosis 2, each and every stuff of meiosis 2 is same as mitosis. Same as mitosis. Everything. But you have one line. Meiosis 2 is initiated immediately after the cytokinesis, usually before the chromosome is fully elongated. There is not proper extension. Before that, meiosis 2 will start. That's why, like normal interphase, here this gap is very short. This gap is very, very short. Everything else is there. Meiosis 2 resemble a normal mitosis. All these steps are same. Don't trade anything. Here in anaphase 2 will be exactly like normal anaphase. Like normal anaphase. Splitting of the centromere and movement of the chromatid. See, like you have one chromosome, then everything like normal enough. Significance of the meiosis, it keep the same number of chromosome in each species, even though there will be reduction, though it is known as reductional division, but ultimately it maintains the same chromosome number 2n in every. And second most important part, increase variation in the population. Uh, I think there were 2-3 points there. summary that we can do later. This done, this done, this done. Majority part is done. Significance of mitosis. Mitosis is popular in the diploid organism, but in some lower plant and social insect, honeybee, even haploid cell can divide by the mitosis. Okay. So why it is important? One thing, genetically cell is exactly same. Growth is growth is due to the mitosis. It maintains nucleus and cytoplasmic ratio. It is responsible for the cell repair, and many cells like epidermis, gut lining, and blood cell will constantly replace because new cell will be formed because of the mitosis. In case of meristematic tissue, whether you take apical meristem or lateral cambium, there is a continuous growth in the plant because of the mitosis process. So I think majority part of this chapter is done. Now you can solve any question by using this last graph. You have first G1 phase, S, G2, prophase, metaphase, anaphase, telophase, cytokinesis, and from G node sometimes cell enter into G1 and then go to S further. But G node, this diagram is not proper because actually what happens, cell exit from G1 and enter into G node. This is what happened like. And we are done with the chapter. How many of you are there? All of you? Jordan, tell me 109. Synaptonymal complex form in which stage and dissolve in which stage? Jordan.
So
Synaptonymal complex form in zygote stage
zygote dissolves
and
in
start
diplotin, yes.
dissolving in diplotin stage.
Diplotin.
This done. Majority of this question done, I guess. Yes, RBC, yes. Sarah, 119. Identify the metaphase 1. How will you identify metaphase stage, Sarah? Sarah, how will you identify metaphase stage?
The chromosomes are like at the equator of the cell.
Correct. So in how many
So
option you have chromosome at equator?
is it option 2?
In option 2 and 3, you have chromosome at equator. This is metaphase 1, not normal metaphase. This is normal metaphase and this is metaphase 1 because pair of chromosomes, not one chromosome, pair of chromosomes. Because we are doing all the questions, let me ask how many of you are there? Pratik, are you there? Chandana,
Yes, sir.
Jordan,
Yes, sir.
Ruhina,
Yes, sir.
Okay. So now we can make it fast. Tuhina, question 123.
Mm, 16, sir.
In metaphase, one chromosome made up of two chromatid. One chromosome, two chromatid. So you have 16 chromosome at metaphase. Diploid chromosome, same 16, because this is diploid cell.
Yes, sir.
Chandana, what change would most directly reduce the duration of the interphase?
So shortened is
Yes, as phase is one of the crucial phase. If this period is short, then duration of interphase will reduce. By the way, we missed one question. Jordan, which of the following, why meiosis is essential?
For asexual, it's not asexual. See, maintenance of chromosome number and across generations.
Yes. Anything we missed from the last topic? No. Grab cycle is done. Tuhina, 126. A cell have 20 bivalent. How many chromosome? Sorry, 10 bivalent. How many chromosome?
Hmm. I think 5, sir. I'm not sure.
20 chromosome. One bivalent. Bi means two. Valent means chromosome.
Oh,
One bivalent
but
have
then
two chromosomes.
somatic cell means.
Normal cell, diploid cell.
Oh, okay, okay. Yes, sir. Yes,
Got
sir.
it?
Yes, I understand. I was having trouble with these kinds of questions.
That's why like discussion will help you to explain key. Bivalent means two chromosome.
Yes, sir.
So if prophase 1 have 20 chromosome, prophase 1 is diploid cell and
Okay.
somatic cell is also diploid cell.
Yes, sir.
Then Sarah, if the spindle fibers shorten unevenly during anaphase, what will be the most? Sarah,
Yeah, sir. Just a second. No, sir, I'm not sure.
chromosome imbalance. Everything else is normal. This is abnormal because you have abnormal uneven space. So only abnormal thing you have in this situation is chromosome imbalance. Chandana, a cell have normal DNA replication but fail to enter into the mitosis due to checkpoint control. Which checkpoint is likely to be activated? Third, why? Example, if you enter into any big giant organization, you will see ki there is checkpoint security guard to check you whether you are right person or not. Same in the cell division, you have checkpoint to check whether whatever process is going on is perfect. Is there any abnormality or mutation? If there is normal DNA replication means S phase is done, then this DNA will go to G2 phase, and after G2 and before mitosis, you have a checkpoint. G to M checkpoint. It will make sure that before entering into the mitosis, cell have everything normal. Any doubt, guys? Jordan.
In the 24 hours average duration of cell cycle of human cell division lasts for about 1 hour.
Yes, less than an hour. 23 are in the interface. So, Hina,
Hmm. So Rana phase 2.
NF phase 2.
Is that correct? Yes, sir.
Homologous chromosomes separate in anaphase 1 and homologous chromosome chromatid separate in anaphase 2.
Yes,
You know,
sir.
full chromosome will separate in anaphase 1 and chromatid in anaphase 2 because anaphase 2 is like normal mitosis. Sarah,
Yes, sir.
Option 3.
yes, NCRT have this thing. If there is karyokinesis but no cytokinesis, there will be multiple multinucleate. Chandana, Chandana,
It's the prophase.
yes. Jordan,
Diplotin, sir, diplotin.
Tuhina.
Sir, is it 4?
In G1, you have two n number of chromosomes. Okay.
Yes, sir.
And when we are talking about M, it means we are talking about mitosis. So if after G1 8, after S chromosome number remains same, it will be 8. After G2, it will be 8. And after mitosis, same number of chromosome, 8.
So 8.
Tell me, any of the question was out of NCRT too difficult, you are not able to do anything from this cell chapter or this cell cycle and division chapter.
No, sir.
But remember, if you want to get full marks in biology, you have to be attentive but calm at the same time. You should be aware what is the demand of the question because you know everything. It's not like you are missing everything. But still, uh, your NEET performance last year was not good because for me, everything is your last year NEET performance. And you have to work on this. How to improve biology? First, you have to accept that you need to work on this. Okay. Objective question like how to make it fast. Second thing is when you do biology, you have to understand question, not just doing the number. Doing the number like I did 50 question, I did 100 question, 200 question, doesn't matter. If you do 50 question with better analysis, by knowing what is the demand of the question, by attempting in the same way like how you attempt in the need to like the more you practice here, you will repeat the same thing. You will duplicate same thing in the exam also. If you are in hurry and doing silly mistake here, you are doing same thing in the exam. Understood? So make sure do it slowly but perfectly. The problem with most of you, you try to make it fast. Okay, I'll answer fast. You will do fast once you are extremely good in the exam. You are making all 90 question perfectly. Only then you go for the speed. Your first preference is the accuracy. Once accuracy reach 100%, then you will look for the speed. How many of you are doing this stuff? Ki you are looking for the speed and accuracy is not 100%. It's okay, even if it is 95%, it's good. But if your accuracy is 95% and you know this stuff, so 90% of 360 will be what? 324, I guess. This is 90%, not 95. 324 is 90% of 360. If your accuracy is, what is the 5% of 360? Maybe around 340. So you are not getting 340 because not that you don't know content, but because you are not perfect in your content, you are not calm and composed, and you are not 100% sure. If you are not making even a single mistake in your practice, there is no possibility that you can make mistake in exam. People who play cricket and play weird shot, they practice it 1,000 times only then they can execute in the exam. So you can't imagine ki if you are doing mistake here, you will do some miracle and do all the question correct. All of you remember this thing and make sure your question is accurate rather than doing all the question. Instead of 90 biology question, you are 100% sure about 80 and have doubt in 10 questions. Don't attempt 10 questions. Give more time to 90, 80 question. If you are perfect in this 80 question, you will get 320. Still your score will be more than 300. But last NEET attempt, you are like you did not get this score. Reason? Accuracy. Accuracy is the key. Understood? So you have two chapters, breathing and exchange, and this locomotion and movement. Small chapter. That's why we can do fast. Take a break. Revise the theory properly. Then I will ask you question. In that case, I don't need to explain you much theory. First, take half an hour break. Don't do anything. Then revise NCRT and understand what is the meaning of every line. Revise again if you did it. Okay. So we finish these two chapters thoroughly. Not thoroughly, few topics are left. We'll revise again. But make sure you revise. Breathing and exchange of gases and locomotion and movement. And don't do any mistake. It's okay doing mistake, but understand how you did this. What was the reason for your mistake? Were you in hurry? Were you unaware about the concept? If you were in hurry, do it slowly. If you were not sure about the concept, read the concept again. Then your thing is done. That's why more than just reading, you have to evaluate every single time how was your paper, how was your test, like how you are doing when I ask question in the class. Are you saying, yes, sir, like are you making mistake and tell another option? Or is it because you are not aware about the answer? Same thing. So tell me one by one, Chandana, what did you understand from this session?